Hello everyone, welcome back. Today we're going to conclude our series on macromolecules with nucleic acids, which are DNA and RNA. So what are nucleic acids? They're basically DNA and RNA. They are the largest molecules in the human body, and since they are polymers, they are going to be made up of monomers. And in this case, these monomers, which are also the building blocks, are going to be made up of nucleotides. So if you see the word nucleotides, no one should say, oh wait, is that a protein? Or wait, is that a carbohydrate? No, nucleotides, you can see the word nucleic acid in the word. And where can we obtain nucleic acids from? We can obtain them from diet, or they can also be made in the human body. So when we mean DNA is actually short for deoxyribonucleic acid, and deoxyribo is a type of sugar, deoxyribose, and RNA stands for ribonucleic acid, where we see the word ribose from. So let's talk a little bit about DNA. So DNA is unique because it holds the genetic information. So basically like the hard drive, the flash drive, you watch those spy shows. I really love those spy movies. The person goes in there, they download their entire information into a flash drive or onto a disk, and that's really all they need, right? They walk out of the building, right? So DNA is kind of like that. All the information the cell has or needs is located in DNA, and DNA is usually found in the nucleus, and the nucleus is almost like another enclosed area inside the cell. And the nucleotides, the building blocks of DNA that we're going to be looking at, are going to be adenine, and they're going to be cytosine, thymine, and guanine. And one unique thing about these is that adenine will always be bonded to thymine, and vice versa. So you're going to see thymine bonded to adenine, and cytosine will always base pair with guanine, or guanine will base pair with cytosine. And you can see it right here, the A for adenine, T for thymine. And when you go back here, you can see that thymine will automatically be bonded to adenine. They would never switch. You would never have a situation where adenine will base pair with the guanine, or guanine will base pair with even thymine. So these pairs are always continuous in the DNA double helix. So how is RNA different from DNA? So first of all, RNA is single-stranded, all right? RNA is single-stranded, and its function is a little bit different than DNA. I like to call RNA kind of like the personal assistant to DNA. It really helps DNA out. Without RNA, most of the functions that DNA is supposed to do, the cell will not be able to do without the help of RNA. So the word ribose is actually a derivative of the word ribonucleic is actually a derivative of the ribose sugar that RNA contains. And here, instead of having thymine, which we see in DNA, this will be replaced with uracil. All right, it's going to be replaced with uracil. So instead of us seeing AT, we're going to see AU, and wherever we have U, it's going to be bonded to A. The same rule will apply for cytosine and then for guanine. All right, so cytosine will always base pair with guanine. Guanine will always pair up with cytosine, and vice versa. So the only unique thing here is that we're not going to see a thymine, and we're only going to see a uracil. So when you start seeing a strand or information that only has U, just know that you're looking at RNA and not DNA. There are also 3 types of RNA. Messenger RNA, which, like the name implies, its job is to help deliver information, and then transfer RNA and ribosomal RNA. I'm going to be talking more about that when we get to the cell cycle, where we're just going to see their function a little bit more. And going back to ribosomal RNA, which is our RNA, where this RNA basically works with ribosomes. Again, when we get to the cell, I'm going to be talking more about this. So this is just a quick recap of DNA and RNA. All right, thank you. See you later.